This week in lab we're going to be focusing on photosynthesis. It's going to be the same setup as last week, except we are going to be using foil because we are going to look at leaves going through photosynthesis and respiration, which you should be familiar with last week. We're going to be looking at either different species or different lights coming through, and you'll see an effect on what that does with the leaves. Photosynthesis is very, very important. Let me get out my pen. Let's quickly show you guys what's happening with this leaf. So we have our sun, okay, and it's giving off sunlight, and so this obviously a plant needs the sun. Your poor plant will not survive in the dark in a closet. It's going to take in CO2. So if you have a plant in your dorm or in your house, always a good idea to talk to your plant. You're giving off CO2. In return, it's going to be giving off oxygen, which is what you need. Okay, it also needs water. If you do not water your plant, if you don't water your plant will be very, very sick and it will die. And in the leaves, okay, it's going to store energy, glucose, okay, in the form of starch. So if you were to put iodine on the leaf, it would turn black. Okay. It's also going to be in what is, if you were to look at a trophic pyramid, okay, we're at the top. These are our consumers. And then down here we have our producers. We're going to talk more about that next year. Producers. Okay, so leaves, plants are at the bottom of the food chain, most abundant, most biomass. So everything feeds off of these plants. Let's dive down into the plants. We know that plants are green due to their chloroplasts. Chloroplasts actually have an outer membrane and an inner membrane. And within that inner membrane, you have the stroma, which is this aqueous solution, which is right here and here. And within that, you have these stacks of thylakoids called the granum. So this entire thing is called a granum, and each disc is called a thylakoid. Within that, you're going to have specific processes. Okay, they're made up of two main processes. Okay, you have the photochemical process, which is called the light reaction, and then you have a biochemical process, and that's going to take place out in this area here, okay, which don't require the use of pigments, and that's going to be called your dark reaction because it doesn't need light energy to take place. Okay. All right, so here are your two reactions. We have our light reaction, okay, and this is going to be your electron transport chain, which I'm sure you guys are all familiar with. Photosystem 2, photosystem 1. The only reason that they are named photosystem 2 and photosystem 1 in that order is that photosystem 2 was discovered second, as opposed to photosystem 1, but it actually happens first. All right, you have your energy from the sun coming in, water coming in. Okay, water is being split, and that splitting of the water is giving off ATP, and then you have oxygen as a byproduct. So oxygen is actually coming from water. That's very important to know. Okay, then you have your dark reactions. These are your dark reactions. This is the Calvin cycle. Okay, CO2 is coming in. CO2 is being 2 units of CO2 can be combined to form rubisco. That's going to go through the electron transport chain. You guys are going to know that in much more detail in lecture. You have byproduct of glucose coming out and energy given off to give energy to your light reactions. So here's a basic summary of the light reactions and the dark reactions. Okay, light reactions, these photochemical reactions, fast, a lot faster than the biochemical dark reactions, but they are light dependent. And again, you're splitting that water to release the oxygen. This is where we're getting our oxygen that we breathe. The main purpose of your dark reactions is to fix that CO2 that it's getting from us, from other animals breathing, into sugar. Okay, and the plant needs that. Okay, and it's being stored as starch, and that's going to fuel the plant's growth. Okay, when you think of a plant, you think of chloroplast. Well, within the chloroplast, you have pigments, and these pigments are going to be absorbing the light energy from the sun, and they're going to start that chain reaction, that electron transport chain. So the most common pigments are chlorophyll A, chlorophyll B, carotenes, and xanthophyll, or xanthophylls. Carotenes are also called carotenoids, and you guys are going to see most of these in lab this week. We're going to be looking at the separation of pigments of a spinach leaf. Now, the one thing I forgot to mention is that chlorophyll A and chlorophyll B, these are your main pigments. Your carotenoids or keratins and your xanthophylls, those are your accessory pigments. And so during most of the season, when the plant, the spring, the summer, the plant has these green leaves here. And it's absorbing the light. Okay. As the seasons change, the night lengthens. Doesn't always depend as much on temperature, but temperature does play a factor, slows processes down. So as the night length increases, this triggers an effect in the tree to slow down that production of chlorophyll A and chlorophyll B. Okay. Then the accessory pigments that are inside the leaf actually start to show their colors. And weather has a huge effect on this. If you have a very wet spring and wet summer, you're going to have lots of pigments. And so as the chlorophyll production slows down, you're going to have a much more vibrant fall than you would if you had a very dry summer. Here we have our photosystem. That's basically just a cluster of the pigments that I just talked about. We have our we have our photon here of energy. So this is our energy that's being coming from the sun. It's going to be sucked up by that pigment. It's going to excite the chlorophyll, and that's going to boost electrons to a higher energy orbital. So think about chemistry, and when you're talking about the electrons and the different orbitals. And this energy is passed on from one pigment molecule to the next. Okay, and as that does that, it goes down its electron transport chain, and it goes from photosystem 2 down to photosystem 1. Okay, I'm not going to go into the exact process of giving off the ADP and NADPH. Again, you're going to be going over that much more in detail in lecture. Alright, so in order to see those pigments, we're going to do something called paper chromatography. That's going to separate them out. So what you guys are going to do is you're going to put a spinach leaf on here, and just a little piece, and you can actually roll up the spinach leaf and just lay it flat on here. You want to make sure that it's not too far down on this point, so you want it right on that line there. And you want to get a quarter, and you want to roll the quarter back and forth to make as much dye as you possibly can. Okay. Now the paper is going to be filter paper, so make sure that you're wearing gloves when you do this, because you don't want the oils on your hands to affect the filter paper. And the paper chromatography is going to separate these pigments out by carrying molecules up the paper with a solvent. So we're going to be using a special chromatography solvent. Okay, and they're going to move depending on their solubility and how well that pigment is able to be absorbed to the paper. And that's going to be based on molecular size, polarity. Okay, so different sized molecules and different polarity of molecules are going to move at different speeds, at different rates. So you have chlorophyll A, you'll see that, and that's a blue-green color. You have chlorophyll B, which is more of a yellow-green. You have your carotenes, which are a yellowish orange, or carotenoids, which are a yellowish orange, and you have xanthophylls, and that's a pretty light yellow. Alright, so your first step, again, you're going to roll up a piece of leaf, spinach leaf. You can also use a maple leaf if you want. If you want to go outside and grab a maple leaf, that's completely fine. Or bring it into class. Whoops. And you're going to run that leaf over along the edge. Okay, you don't want pigment to be down here. Okay, that's bad. So let me erase that. Pigment is bad down there. You want all your pigment to be right here. Okay, and if it's a little bit higher, that's fine, but you don't want it down there. The reason why is that you are going to be sticking this in solvent, and if the solvent touches your pigment, it's not going to run. You actually want to have this solvent run up the strip, and it's going to run up over a course of about half an hour, 45 minutes. So you want to do this step first, and as it's running up, it's going to be separating out those pigments. Okay, make sure that you get a lot of pigment on the pencil. It's going to be 1 ml of solvent at the bottom. Okay, you can hang it from the hook, so they will have these little hooks here, and you'll have the filter paper. Okay, you can hang it from the hook, or you can just place it in. You make sure that you cork it because you don't want those fumes escaping. And you want to wait, takes about half an hour, 45 minutes, until the solvent reaches about 1 centimeter from the top. You're going to be able to see this due to a solvent line. So in the previous picture you would have seen a solvent line, and which would be probably about here. And then you know that's when you're going to stop it, you would take it out, and then you would measure. So we are going to be calculating the RF, and basically the RF is just a relationship between the distance that the pigment has moved and overall the distance that the solvent has moved. And this is a standard number, so if you were to do this 50 times, you would get a pretty standard number for that, for either chlorophyll A or chlorophyll B. It is a constant. Okay, it depends on how much pigment you actually put, so you would have to put a standard amount to get that standard constant number. And it's pretty easy to calculate, just basically distance moved by pigment over distance of the original solvent. Now we went over this last week. Just want to quickly remind you again, oil rig, so oxidation is to lose electrons, where reduction is to gain electrons. And a lot of people get that confused. Even though something's being reduced, it's gaining electrons. So water is losing electrons, it's being oxidized and going to O2. If you remember from respiration, the opposite happened. So during respiration, water was being reduced, or oxygen was being reduced to water. And this is what your setup is going to look like. It is the same thing that you had last week, except that you have leaves, and leaves are going to coat the bottom of your chamber. You're going to be measuring CO2 consumption and oxygen production. This is the opposite of last week, where you would have had CO2 production and oxygen consumption. Okay. We're going to be looking at respiration by putting the chamber in foil, waiting 5 minutes for everything to kind of slow down and switch, and then recording to see how the leaves go through respiration. When we look at photosynthesis, we're actually going to be, I'm going to move this down here, 1 second. So when you guys have your setup, you're going to have your chamber here, and you're going to have your one probe and your other probe here. Okay. When you're looking at the photosynthesis, whether it's going to be with a film that you're going to put over, so a green film or a red film, or if it's just plain, you're going to have a heat sink. And that's just going to be a bottle filled with water, and then you're going to have your lamp. And the lamp is going to shine light through the bottle so that you're going to get light and heat. That's supposed to be heat there. This is going to trap the heat, and so that all that comes out is just light. Okay, so make sure that you have a bottle filled up with water. That's going to be considered your heat sink. And then we're going to determine the rate of respiration and photosynthesis, just like you guys determined it last week, where it was just respiration. You should have 3 runs. You should have a positive control, and you should all know how to figure that out. You should have a negative control, and you should all know how to figure that out. And then you should have your unknown sample. So your unknown, this is what you're going to be comparing to your positive and your negative control. You could either use the filtered light, so the green film or the red film, or you can change the species. So originally we are going to use spinach. You can do spinach versus maple leaf. You could do spinach versus a holly leaf or something else. We could bring lettuce in, whatever you want to bring. So if you have lettuce, it's lunchtime, you want to bring that in and you want to compare spinach to lettuce, that's completely fine. Some tips. Make sure that you air out the chambers between runs. I know a couple of people didn't do that last week. That could have been a huge error. Also, if your spinach leaves are wet or if there's any water within your chamber, that's going to give off oxygen that's going to completely mess up your results. Make sure that you're waiting 4 to 5 minutes between runs, and that's after the sample has been placed in the chamber. So if you're starting with respiration and you've got that chamber and you've covered it in foil, wait 4 minutes, and then you're going to press play, and that's a big thing. Make sure that you press the play button. I know a lot of people didn't do that last week. Then it's going to run for 10 minutes, and then you can take your readings. Make sure that you have that button of the filing cabinet to store your data. You're not going to do file new or file save. You're going to store it. And then you're going to start again. So you're going to add the leaves, add the film or no film, and you're going to wait 4 minutes because it's not going to switch instantly from that respiration to photosynthesis. You got to give it some time. So about 4 to 5 minutes, then you're going to press play. The sample is going to run for 10 minutes at that 2 samples per minute. Okay. Last thing, you're going to fill out the data sheet that you have in your lab notebook. I am also adding an assignment onto this. So this is really important for you guys to know. Your raw data that you are collecting will be posted on Moodle. So when you guys are all done, you're going to bring your lab quests up. I'm going to put it on my computer, and then I'm going to post it as a TXT file. That means that it can be opened up. It's just a text file. It can be opened up either in Excel or Numbers. So if some of you have Macs, you can open it up in Numbers. If some of you have Dells or PCs, then you can open it up in Word. In Excel. Okay. You're going to make a graph, and I will quickly do a video on how to make a graph with using your group. But because you're using your group's data, I want you guys to make your own graph. I want everybody to get used to how to make a graph. Your graph should have axes, so make sure you know what's here and what's here. It should have a legend. Okay, so if you have lines going like this, you want to make sure that you are labeling what these lines are. Okay, and then you want to make sure that you have either a figure description or a title. And you're going to be making 2, one for CO2 and one for O2. O2 doesn't always work, so if we get some really bad data, then we may not be posting O2, but for right now, you're going to be making 1 for CO2, and it's going to have your 3 runs on there, and 1 for O2. So this one would be for O2 or CO2, and then you're going to have some lines going down. Okay? So make sure that you are posting both of these graphs onto Moodle. I'm going to make a forum. And again, graphs should be your own. And the last thing, make sure that you are labeling your light and dark graphs. So basically, which one is going to represent photosynthesis, which one's going to represent respiration. Okay, photosynthesis, don't confuse light and dark graphs for light and dark reactions. It is not the same thing. Okay, so don't say this is the light reaction, this is the dark reaction. We are not specifically looking at these two reactions. We are looking at photosynthesis versus respiration. All right, I hope you guys have a good week. There will be 3 clicker questions when you walk in, so make sure that you are prepared. I will see you guys all next week. All right.